Welcome everyone. We are doing another BGCSE past paper. Today we are doing a biology paper. It's an alternative to practical paper, which is a paper 5. And this was from the year 2019. Before you start the examination, always make sure that you write your school number, your candidate number, your surname, and your initials. Also, as I always encourage you to do, is to read through the instructions carefully to make sure you understand all instructions before you start the examination. If in doubt, please refer to your examiner or direct all queries to your examiner before the start of the examination. All right, so this, again, paper 5 for biology is a 4-question paper that are related to your experiments or practical in biology. And normally, you have to write up a lab procedure. So you'd observe a situation and write up a lab procedure. So for question number 1, let's dive into this one real quick. And for question number 1, it's a diagram shows a light microscope with a very simple microscope. All right, and let's see what they say. Firstly, there's a use label lines to label the following parts of the diagram or on the diagram. What I'm gonna do, they only give us 3 parts right here for 3 marks, but let's just label as much as we possibly can. All right, and so this piece up here is called the eyepiece, right? So let's do that real quick. That's the eyepiece. All right, and I said that's the eyepiece right there. All right, the eyepiece, matter of fact, it's called eyepiece lens, or you may want to call it ocular lens. Okay, so it's also called ocular lens. All right, so that's the name of that part. Now you have this part here is another type of lens, and this is called objective lens. So let's label this as the objective lens. So the objective lens. And remember they say use label lines, so you should use lines, right? When you're doing it. And this part right here is called the nose piece. So this, I'm gonna put it right there. Right there is called the nose piece. Perfect. All right, so that's the nose piece. All right. We also have right here what is called the stage. So the stage is this platform portion. So this is called the stage of the microscope. And then we have the diaphragm, which is right here. The diaphragm is going to be right here. All right, so that's the diaphragm. And on the diaphragm, what you're going to see on the diaphragm are some small knobs, and some microscope is some small knobs. Those are what control the diaphragm or the rotation of the diaphragm, right? All right, so the diaphragm, and these are the knobs that you'll see on the diaphragm. A different microscope will have them oriented differently. So this yellow portion I'm putting here, those actually help to adjust the diaphragm and also the clip on the stage for this type of microscope. All right, another name, the mirror. So this is the mirror right here. All right, so that's the mirror right there. And then let's go a little bit down. We have the whole thing right here. This part is called the base. I'll put the base and put the name right in it. That's the base right there. Okay. And then we have this portion is called the arm. I think I could put it right in it. So this is called the arm. All right. So that is arm is like an handle. All right. So that like the handle that you're going to use to carry it. When I see what else could I label? Okay, yeah. So this block knob, let me put the name over here, and then I put a line to actually indicate it. So we have up top there is the course adjustment knob. So we're gonna have what they call the course adjustment knob. Okay, and then we have the fine adjustment knob at the bottom right here. So we're gonna have a fine adjustment knob. All right, fine adjustment knob. All right, and so just quickly let's label those. So these are the knobs. All right. So that's one there, this one right here. All right, so those are the 2 knobs to adjust the focus of the different lens. So they help to adjust the focus of the different lens. All right, so let's go down to the next part of this question now. All right, so we already labeled those 3 parts. All right, so now it asks us now to state the function of the mirror on that microscope. So the function of the mirror is simply to reflect light onto the stage. So it's going to say to reflect light onto the stage. or on the specimen, or on the specimen, right? And the specimen is what you're actually observing on the specimen on the stage. All right, so it's really to really focus light towards the stage and to at least to make you see the specimen much better. All right, because once light is there, you're going to see the different parts of a specimen way, way, way better. So for part C, now, if a specimen is viewed down the microscope using an eyepiece lens with magnifies x4, which means times 4, and an objective lens, which magnifies times 10, what is the overall magnification for the specimen? So the eyepiece with a magnification of times 4, and then the objective lens with a times 10. So the first thing going to happen is that the one going to multiply it by 4 first. So you're going to increase the magnification by 4, then you're going to further increase by another by 10. So increase by 4, then by 10 is going to be 10 times 4. And so the total magnification is going to be 40. 40. All right, so it's going to be 40 times. All right, so equal to 40, or you can just write it as this x 40. So it's times 40. All right, so the magnification is times 40. All right, next part of the question. Here now it said that the diagrams show the steps involved in making a wet mount slide for viewing down the microscope. So it is notice the keywords they're using here. You're actually making a wet mount slide. All right, so the slide going to be moist. And normally moist slides are better to actually view for different reasons. All right, so we'll talk about that if they ask us. All right, so which type of cell is most likely to more likely being placed on the slide in step 1? So let's go to step 1 right here. All right, so in step 1, what is important here, you notice the person actually swabbing the mouth. All right, so what type of cell is normally going to put on this based on the action right here, and we can call that cheek cell. So normally you take cheek cell from your mouth. So the more likely cell going to come out here is cheek cell, okay? The cheek cells. All right, so next part now says stain is being added to the slide in step 2. So you put stain to the slide in step 2. It's a named apparatus being used to add the stain in step 2. So what is the name of this apparatus? And it's called a dropper. A motherfucker, it's called a pipette dropper. So it's a pipette dropper. Or you can just say the dropper, whichever one, it don't matter. I think they will give you the mark for it, all right? So it's a dropper. You can just say dropper or pipette dropper. Really don't matter. Saying the same thing, right? All right, the next part of this now is why must stain be added? So why do you want to add stain to the specimen? And it really to make the parts or the structure of the cell more visible. So you actually get a better viewing of the cell itself. All right, so it's to make the parts, all right, or structures, let's say parts or structures, all right, of the cell, of the cells, more visible. Yeah? So that's the reason why I actually do that. All right, so that's 1 mark. For F, part F, it says step 3 shows how a student slowly lowers the cover slide or cover slip on the specimen. Why must the cover slip be lowered slowly? So why you want to put it down slowly? All right, up by beside being obvious, you will break the stuff if you really do it hard, right? Well, that's not the reason. I'm just kidding. But yeah, you could break it, of course, because they're glass, right? But the true reason why I want to lower it is to reduce the possibility of bubbles between the 2 slides, okay? So to reduce air bubbles, all right, from being trapped, between the slides. All right, between the slides. If you push it down too hard, the bubbles will be between the slides. And of course, looking through the microscope and look at the bubbles, you may even confuse parts of a cell with the bubbles as well. So you want to make sure that you have little to no bubbles between the slides. All right, so the last part of question number 1 is to state the function of the cover slip. So the function of a cover slip is to prevent the specimen from drying out. Okay, so keep moistures in. All right, so it prevents the specimen from drying out. Okay, from drying out. All right, so in other words, it keeps in moisture. Let me just say, so the cover slip keeps in moisture. All right, and I said earlier that moisture is very important to get a better viewing. When the cell is dry, some of the parts may not look as nice or well defined because when cells lose water, they become flaccid and the structures tend to change. But the cells are turgid in a nice natural form. It's better to see the structures. All right, especially if you're looking at living cells and looking at cells carrying a certain function. Like let's say looking at mitosis or meiosis in the cell. Then of course when the cell is moist, it's better to see it, but on a dry cell. Matter of fact, the cell is dry, gonna die. And so some of the parts may not look as the typical shape or the typical orientation and so on. All right, so you may even confuse cell parts based on if they're dry. Some of the parts may not even be seen, to be honest. All right, for question number 2 now, right? You see, a student carried out an investigation on osmosis by submerging potato strip in 3 different solutions. And it seems as if paper 5 tend to carry a lot of osmosis question. All right, so if you notice, this is not the first osmosis question we're seeing here. It's not the first on potato. And I guarantee you're going to see it again, right? So for part A now, say all strips were cut to the same length as shown. I say measure the length of the potato strip in a suitable unit. All right, and record it. So this slip, this I already measured this already. All right, so what I got from my ruler by measuring this, and this is to scale, just to make a note of that. That's why they say measure it. And so what I got is 3 centimeters. So you can use 3 centimeters, or you can say 30 ml. So whichever one is good. Because remember I said use a suitable unit. What they do not want you to use is inches. They do not want you to use inches. So either you use centimeters or you use millimeters. All right, so those are appropriate units used to measure in science, particularly biology. All right, inches is not necessarily used as a standard measurement or a common measurement. All right, so part B now, we say after 24 hours, the strips were removed from the solution, blot dry, and their new lengths were measured, right? Now for strip A, B, and C, notice they look different. All right. Now it said measure and record the length of the 3 strips and record your results in a suitable format in the space below. All right, so I should have drawn a table. All right, I'm gonna skip that part out, but I'm just gonna show you this real quick. What I need to do first is to draw a table, right? So we're gonna have a table. You know what? All right, because I want this a little bit neater. All right, what I'm gonna do is to write the headings first, then I try to put my table in, and then I show you exactly what you should have. All right, you can have at least 3 things, but it's 4 marks, so you want to have at least 3 columns. 4 columns is also good, but I'm gonna show you what you can do here. So the first column, we're gonna put the strip, all right? So I'll put the strips here on the first column. This will be our strips. And then my second column now will be my original length. I want my original length. That's going to be important. All right, so original length. All right. And I want my final length as well. Matter of fact, you know what you need to do? Make sure you put your units as well. So this is going to be, let's put millimeters, all right? You can put centimeters as well, not a problem. And let's look at our final length now. This will be our final length. All right, and then let's put again the unit, millimeters. And let's say another column here, going to say change in length, okay? Change in length. And I want to point out something here. Change in length, again, that's going to be in millimeters. All right, millimeters. I'm going to put one other column. I'm going to call it percentage change. All right, let me bring these over a little bit so I have more space to play with on the end. All right, so yeah, I think that should be okay. All right, yeah. And then the final thing, I'm going to have a percentage change. I'm just going to put symbol percentage change in length as a percentage change in length. All right, and again, that will be in percentage. All right, so once I have that now, I could put my strips in, right? So I'm going to have strip A, B, and C. So let's say this is A. Then I'm going to have, let's put B right there. And then let's put C there. All right, and what I'm going to do is also put columns, but let me put the columns last. I just want to kind of do this for you. All right, so the original length of all of them, remember we measured the thing at 30 millimeters, so all of them will be 30. So let's put the 30 for all here. 30 ml, millimeter, I should say. I said ml, yeah, millimeter. All right, so the next one here, yeah, let's put this across. That will be 30 as well. The next one now also will be 30. All right, and so let's go into our next column, which is our final length. And so I already measured these. So strip A is the shortest one, if you notice it, and then strip B and strip C. All right, so I already measured these. I'm gonna plug them in. The first strip, the final length, which is this, is actually 24. 24. And then the next one. Now let's go to the next one. It is 30. So that one is same. And the last one with the strip C, that one is 36. So this is 36 here. All right. The change in length, which is the difference in the length. Now, the difference between 24 and 30, that is 6. You can also put -6, not a problem, because a minus will tell that a decrease in length, right? So it's decreased, so you can just put -6, but as a change, it's just going to be 6 for me. 24 minus 30. But here, the change in the strip B is going to be 0. And then the change in C is going to be 6. So this one is a positive 6, right? Mean increasing. So a -6 means that is a decreasing, and a +6 means that is an increasing. All right, so you can put a negative sign in it if you want. You don't have to. All right. All right, but you can also make a key that negative means decrease and so on. All right, so you can also have a key below the chart. In the final column now, in percentage change, the percentage change is a difference over the original. So to find the percentage change, I'm just work out one, and I don't need to work out all of them. So to find the percentage change for the first strip, you're going to say 6, which is the difference, divided by the original, which is the 30, and all of that will be multiplied by 100%, right? So once you do that, you multiply that by 100%, so times 100. All right, so once you get that, all right, let's say I could move this, I think so. All right, let me just try to move this. All right. All right, so let me see if I could move this. All right, I'm not going to get to do it. Okay, that's fine. All right, so this was multiplied by 100, right? I don't like it look, but that's the least. Once you understand exactly what I'm doing, you should be okay. All right, so let's put the next one. Let's get the next one in. All right, don't, in interest of time as well. All right, I'm doing something wrong, it seems. Let me see something. All right, I am good now, I think. Yeah. All right, so the next one here now, it will be 0 over 30 times 100. That is also 0, so this 0% right here. That percentage change is 0. And for the last one, it is 6 over 30, which gives us 20. So it's going to be 20%. Or matter of fact, the top one equals to 20% as well, right? So that's 20%. That's the work out one. So to work out the percentage change is always the difference over the original, and then you multiply by 100%. All right, so that's how you actually work it out. All right, so yeah, so we're good with that. All right, so let's go to the next question now. All right, our next part of question. It said complete the table below to describe the expected texture of the potato strips A and C after being removed from the solutions. So if you look at A, again, let us go back and tell what happened to A and what happened to C. If you look at the chart, the chart will also tell you. A decreased and C increased in length, right? And please not say potato grow or the strips grow. They're not growing. I've seen that so often and really is not correct, because to grow means to produce new cells, right? It's just like expansion. So you get longer, it means that it expand or increase in length. So not so the strip is growing. So here now for A, again, because it gets shorter and smaller, that means it lost water. And so as a result, we can say this one becomes what? Flaccid. All right, for the next one, strip C actually increase in length. So there's an increase in length. We can say this one became turgid. All right, so the texture is flaccid, turgid. When it increases, turgid. When it decreases, it's going to be what? Flaccid. All right, so that's that for that part. Now for part D, in this question here now, it said identify which strip was placed in distilled water and explain your choice. So again, even coming from the chart just that we just did, we said strip C increased in length and became turgid. So therefore, strip C is the one that placed in distilled water. And in this case, you can say that the solution or the condition is hypotonic. Okay, so I'm going to make a note right here. The same effect, distilled water is the same effect as hypotonic. Okay, so hypotonic and distilled water give you the same effect, hypotonic. All right, so the answer you're going to be strip C. Okay, so strip C. And why is strip C is because simply, and it said explain why, right? So simple because there's an increase in length. So there is an increase in length. There is an increase in length, which indicate, let me say, increase in length. All right, the increase in length, which indicates that water enters the cell, all right, causing as I say, enters the cells, all right, causing the strip to become turgid. Okay, yeah. So they cause the cell to become the strip to become turgid. So that's how we know for sure that it is distilled water, because distilled water outside of a strip would have a higher concentration of water compared to the inside. So therefore, water move from the outside to the inside, causing the strip to become bigger or longer and also turgid. And you can also explain further that this is caused by water entering the cell from a higher water concentration to a lower water concentration through the membranes of the cell within the potato strip. All right, so just to make a note of that. So just to explain right here. So water enter from the outside of the strip, and I put in here higher water potential or higher water concentration. So higher water potential or concentration. Okay, water potential or concentration to the inside of the strip where there is less water, and less water mean lower water potential. Okay, all right, through the membranes of the cells, through the membranes of the cells in the strip. All right, and you don't need all of this to get those 2 marks. I'm just kind of giving you some extra information if you need to explain further. Yeah, but this is what exactly I mean in a hypotonic situation. Water leave from outside to the inside cause the cell to increase in length, and that increase in length now will cause the cell to become turgid, and also in that case as well, the cell membrane will move towards the cell wall, all right, and the vacuole will also increase, and the turgor pressure within the cell will also increase as well. All right, so last part of this question, we said compare the concentration of the solution that potato B was placed in with the concentration inside the potato strip. And remember what B was. B, let me go back to the chart and show you real quick. For B, there was no change, and so therefore the percentage change is also 0. And because of this, because there's no net movement of water, the water that was going in equal to water coming out. And simply because both are the same concentration, and so therefore is what they call a hypertonic concentration. So right here now we say the concentrations, compare the concentration. So therefore the concentrations, they are the same. All right, so the concentrations, all right, are the same. So they're the same concentration inside and outside. And so I'm putting in bracket here, this is called an isotonic solution. So the solution outside of the cell is called isotonic. All right, the isotonic solution. All right, mean equal concentration. So what going in the cell equals what coming out of the cell. All right, so the concentration outside of the cell and inside of the cell are the same. All right, so question number 3, and we have one more question to do on this one. It says a mother kept a record of her daughter's height, all right, from the time she was born until she was 8 years old. The data is shown in the table below, and so we have the data right here. And it's until she is 8. So let's go down a little bit more. All right, so 8 is right there. Cool. So right here we have the age in years. Let's go back a little bit more so make it better for you to see. Yeah. All right, good. So we have the age in years and we have the height in centimeters. And then notice 0 to 8. And then we have 42 all the way to 128. And then now it asks us to do something. So let's see what they're going to ask us to do here. And it said draw a line graph of the data shown in the table. Use a ruler to join each point to the next. All right, so what I did now, you're going to see a copy of this. So a copy of the graph is this here. So I have a copy of the graph. Let me just go in a little bit more, zoom in a little bit more. This wanted to see this quite well. All right, so there are certain things I want to point out when it comes on to the graph. One, you must use appropriate scales for your axis. Very important. The scales must be appropriate. Why? You want a graph to be at least half of the page. Not, if a graph is very, very small, then you're going to lose mark for it. All right. Also, you want to make sure that you label both axes as well. Do not just put on numbers and don't label. You have to label the axis as label them and even put their units there. For example, height is measured in centimeters based on the chart. Number of years or the age in years. All right. And you plot a graph looks something like this. Also, what I want to point out that you need to make your points or your coordinates very smooth and pointy. And your lines supposed to be really, really smooth as well. Okay, your points must be really smooth. Your graph will shape something like this. All right? So, just save some time. I already done it. So, you can look at this and see, okay, if your graph will be the similar thing. But the idea is you should know the plot graph. And almost all of these paper 5, they do have graphs for you to plot. All right? At least one graph. All right, so bear that in mind. So all paper 5, based on what I'm seeing so far, is there's a graph question for the most part, and also you need to write up a procedure for some experiment. All right. Obviously, you have to do observation as well for all possible experiment. All right, so that's the graph there. Let's jump on to the next part question. All right, and dive into it as much as we possibly can. All right, so next part of question here now, which is part B of this question, and it states here that state between which 2 years the child height increased the most and increases the least. So we're looking for the increase the most and increase the least with 2 years. All right, so based on the graph itself, you see them on the graph, and you can, motherfucker, you can't even go back on the chart and actually see it, right? Between the different ages. So the one that increased the most is between 1 and 2. If you look at 1 and 2, difference here is 36, right? So between age 1 and 2 is a big jump by 36. The others are really smaller than 30. If you look at them, all right, very small differences going down like that. So the biggest one here is actually 1 and 2. So let's actually put that in now. Ages 1 and 2. I said it's going to be 1 and 2 here. 1 and 2. All right, and say you can say ages 1 and 2, and it said increases the least. And so if you look at the chart again, you're going to realize that the smallest incremental change here, and again you can look at the graph, you actually see it, or you can look at the chart, you actually see it. So difference is 7 and 8 because here is 24 to 28. That's us 4 compared to the others, right? All right, so yeah, we're going to have between 7 and 8 is the least. All right, and I guess this was practice again. I mean, just to know, okay, look for certain things on the graph and so on, right? So the 7 and 8. 7 and 8. All right. So those were the different most and least increase in terms of height. All right, this is part C. Said describe the growth pattern shown by the trend of the graph. And look at the graph carefully. You're going to notice a number of things. So let's go back to the graph again one last time. I notice the pattern of the graph is a general increase. Okay, so there's a general increase straight across the board. But of course, at different point, you notice there's something happening here. All right, so I'm going to mention it. Notice the graphs have a slope almost coming to a straight plateau type of shape. It's not totally plateau, right? It's not plateau at all, but it's look like it's just kind of tapering off. So there's something happening there, right? So we're gonna make note of that. So one point that you can get here, the first point that you can actually make is that the growth generally increase with age. Okay, so growth. So once you're growing or once you're getting older, so growth generally increase with age. Okay, so as you get older, yeah, you get taller. And next point that I can make as well as that after age 5, so let's put that one here again as a next point. After age 5, all right, based on what we're seeing on the graph, after age 5, the rate, or so the rate of increase, there's still an increase, but the rate of increase slower, all right, is slower compared, let's say this compared to previous years. All right. So after age 5, the height is getting you're still increasing, but increasing at a slower rate compared to the previous years. All right. So we can say it a different way. Before age 5, the height is rapid compared to after age 5, right? So you can say it in different ways. Now the last part here, part D, is a state 2 factors that can affect the growth rate in a child. So what are some of the things can affect growth rate in a child other than age? So beside the age, what causes it to grow differently? And of course, one could be diet, because it depends on how you're eating, what you're eating and all that. Then you may grow faster than the other child, right? So if you have a protein rich diet for the child, obviously the child going to make more cells faster because of greater protein, and grow faster than a child that is malnourished or not getting enough proteins or other nutrients. And so for this reason, to feed our children, right? Not too much now, not excessively feed our children, but feed them well to make them healthy and nice and grow appropriately. So the diet or nutrients, all right? So it could be diet / nutrients. Let's say nutrients, particularly our macronutrients such as our proteins and so on. So diet / nutrients. It can also be on your genes as well. Genetics could play a role in this as well. So it can be genes or genetics or heredity. All right. And for that, because remember now, there are some persons that are short, naturally short, based on the family background or the inherited trait from the parents. And if you're naturally short, you're going to go tall for a period of time. You're going to be really, really stuck in a certain height. All right, and your growth rate also going to be slower. All right, so genetics definitely could play a role in that. Gender also, boys versus girls, and generally girls will tend to grow faster when they are younger than boys, right? All right, and the next one could be here is also hormones as well. But hormones is let me put it still. It could work, but the 3 one on top will be the best ones. All right. So if there's hormonal problems, let's say you're producing a lot of the growth hormone, then yes, you're gonna grow excessively. You can have hormonal problem cause it to grow really, really tall, or you're not producing hormone that not effectively producing the hormone, and so your growth rate will be slower or stunted. Right. All right. So that's end of that question, and we're at our final question now. Question number 4. It said diagrams show materials used to investigate the action of catalase on a strip of raw and cooked potato. Now, I want to talk about catalase a little bit. Catalase is an enzyme. Matter of fact, once you see N with the ASE, you know it's an enzyme. And so catalase is found in living tissues, such as the skin. If you cut the skin, you get catalase from there. You get it in liver, you get it in potato. So anything that is living contains catalase. Now, what catalase does is to break down hydrogen peroxide and they produce water and oxygen, right? So catalase can break down hydrogen peroxide to produce oxygen and water. And that's why when you get a cut and you pour hydrogen peroxide on the cut, you see form bubbles. The bubbles is because there's an excessive production of oxygen that's escaping the water liquid. So therefore you get bubbles forming. So bubbles are due to the presence of what? Oxygen from hydrogen peroxide when it reacts with catalase. And catalase is present in all living tissues. All right? So here now, we have raw potato, we have our cooked potato, we have our graduated cylinder, we have our test tube, we have our test tube rock. Yeah. All right. So here now it asks us to say use the apparatus shown to write a step-by-step method to investigate which potato strip has more catalase. All right. So let me just quickly run through the steps that you can make right here. Right. So the first one, and it's something you need to practice writing. Matter of fact, not even just writing, but do it all the time. You should always try to do this all the time. Clean all apparatus. That's the first thing. Clean all apparatus. Make sure you do that. Yeah? All right. So it just being cleansing, isn't it? Yeah? Like that lab clean and nice. Yeah. So cleanliness is next to godliness. Right. And I don't like the devil, so I don't like nothing dirty. So make sure you clean your stuff. Make sure everything is looking nice and clean. It's not because of appearance, really. It's really because of to prevent contamination, and also in some cases to make the apparatus work at its optimal best. Especially a thermometer, you clean the bulb to make sure that you read the temperature correctly and so on. All right, so cleaning is very important for the lab. Prevent contamination, and it's good practice. So when you become doctors and so on, and you're in the surgical room, you make sure your room is sanitized and properly cleaned so you don't cause the patient to die because of infection. All right? So, all right, so that's good practice. And it's just being the right thing to do. It's the right thing to do. The opposite is bad. All right, so second step is to place the test tubes. So let's place the test tubes. Place the test tubes. The test tubes in the test tube rock. In the test tube rock. All right, so you're going to do that. Place them in the rock. That's the next thing you want to do. All right. And the next thing you can do is to, matter of fact, let's say label. Let's say label and place the test tubes in the test tube rock. Let's say label. That's very important. Label and place the test tubes in the test tube rock. Right. So very important. But to label stuff. Always try to label. Labeling is important. So you don't make mistakes. You don't mix up which one is which one. You know exactly, okay, this was the cooked one. That was the uncooked one. And you can use codes if you want to when you're labeling. You don't need to write out the whole word. You can write ARB, 1, 1, 2, things like that. All right? So after you do that now, and you place the test tube in test tube rock, what you want to do now is to place potato strip in each test tube. Okay? You're going to place a strip of potato in each test tube. All right, and you can make a note that one will contain the raw potato and one will contain the cooked potato. All right, so you want to place them in the test tube. All right, so place a strip of potato in each test tube. All right, that's our third step. And this is a 6-mark thing. All right, so 4, even though we're going to end up with more than 6 steps though. Because I'm separating the steps. You can merge some of the steps together if you want. All right, but it's a simple 6-mark. So the fourth thing you want to do is to measure the specific volume. Did I measure the strips of potato? No, we don't need to measure them because the potato strip must be the same. Okay, just make a note of that. You must make sure the potato strips the same length and the same size. All right, we're not going to take the measurement of the length at all, but need to make them the same size for fairness in the experiment to make sure this experiment is accurate. All right. So the fourth one now is to measure a specific volume of hydrogen peroxide. We're going to measure a specific volume of hydrogen peroxide. All right, so we're going to do that. And you can decide how much you want. So let's say you can say 4 ml, 20 ml, whatever. You know, you decide. Let's say 6 ml. All right, so measure 6 ml. All right, so that will be our 6 ml there. You can specify the amount you want. You can use a little bit more, a little bit less, depending on what you want to do. It don't matter. But if the test tube is small, then you cannot use too much hydrogen peroxide because it will leave no space for the bubbles to form. The bubbles will keep running over the test tube, right? So our next step here now is to pour the hydrogen peroxide in one of the test tube. So I'm not gonna say in two. I'm gonna make sure to do this one at a time. So pour the measured. Let's say pour the measured hydrogen peroxide in one of the test tube. In one. Make sure you say in one of the test tube. All right, not in both of them, but in one. You do it one at a time. One of the test tube. All right. Of the test tube, and then I'm gonna bring a next thing right here now. Allow the let's say I'm gonna put this now. Allow it to sit. Yeah, allow it to sit at a specific time. I could do that. Yeah, that makes sense. Allow the apparatus, which I'm talking about the test tube, that allow the apparatus to sit for a specific time. And you can say, okay, for 20 seconds, you're going to make it sit only for 20 seconds or a minute, or, well, a minute will be too long, to be honest with you, because the reaction will take place fast. So let's say 20 seconds, right? You don't really want this to go too long. If it stay too long, you're not going to get the accurate measurement of the bubbles. You're going to just bubble, and then the bubbles will die out after a while, or it will run over the test tube, right? All right, so this is now our sixth step now. And it's what you can do is to measure the height of foam that is formed. So we're going to now measure the height of foam formed. Okay, so measure that of foam formed. You're going to make using the ruler. All right, a foam formed using the ruler. I can simply say using the ruler to measure that of foam forms. You can even do it that way. You can say use the ruler, or by using the ruler, measure the height of foam formed. And let me give you one more step after measure the height of foam formed, which is 7. You're going to say repeat. So notice I only said one test tube earlier, right? So you're going to repeat now. Repeat steps. Where did I start this from? Measure the, okay, yeah, from step 5. So repeat step 5. Yep. Repeat step 5 to 6, or step 5 and 6. All right. Or you can say measure step 4 to 6. Yeah. Repeat steps 4 to 6. All right. For the other test tube. For the other test tube. And the reason why I want to do them separately, because remember that foam will form quickly. And so you don't want to have the two test tube and then you measure one and then go back to measure the next one. No, you want to make sure you give them the same accurate time, the same amount of hydrogen peroxide, and so you get a better control of what you're doing rather than trying to do all of them at the same time. You may end up in problems. And of course, you're going to end up measuring one before the other one. But you want to make sure you measure them in the same exact amount of time. All right, and so repeat the steps, and then you're going to say record the results and record all results. So you're going to say record all the results. All right, and of course you can make calculation, the predictions. Yet, if you want to make it a little better, nice it up a little bit, you can say, yeah, use the recorded results to analyze the data to whatever it is, the whatever, blah, 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 and make observations and all that. All right, but those are the basic steps you can have there, all right? And that pretty much those steps will kind of guide you through most of the labs that you need to do, you follow certain safety rules, and that's the next thing you put in as well. Make sure you follow all the safety rules, make sure you do that, make sure you do that. So when you write up this paper 5, and because I'm telling you, paper 5 always, based on what I've seen so far, is asking you to do up a step. So remember to include safety rules, remember to include clean up the apparatus, remember make sure the workstation is organized, make sure you have all equipment, things like that. So you need to put in those when appropriate, okay? So you need to look carefully, read the thing carefully, see what they give to you, and then if you need to include the additional stuff, please ensure to do so. All right, so for part B now, part B1 is to describe the expected result of this investigation. Now remember, we have raw potato and cooked potato, right? All right, so we have raw potato and cooked potato, and the catalase is an enzyme. So in the raw potato, because again, catalase is found in living tissue, so the raw potato, will have catalase, and so what you expect to see is bubbles forming in this. All right, so you're gonna see bubbles formed formed readily. Let's say readily or easily, or a lot of bubbles will form, or bubbles form immediately. All right, so you will see bubbles. Once you have the formation of bubbles, you're gonna get the mark. I could tell you that. Now, for cooked potato, for cooked potato, what you will see is no bubbles. So this will be no bubbles at all. And you must know why by now, why there be no bubbles, right? Remember, catalase is an enzyme. And if you cook the potato, what happened to catalase? They destroyed, they denatured. So explain the expected result described in B1. All right, and so this is where you're going to explain it now. You're going to say that the catalase will work in the raw potato because it's found in living tissue. So let's say catalase. The catalase is found in living tissue, all right, in living tissue, and will be present in the raw potato. Will be present in the raw potato. Okay, that's very important right there. All right, but is denatured. But is denatured by cooking. Okay, but it's denatured by cooking and will not be active in the cooked potato. All right. All right. In the cooked potato. All right. Hence, no bubbles form in the cooked potato. Okay. So hence, no bubbles. All right. No bubbles will be formed in the cooked potato. All right. So see, yeah, easy 1, 2, 3. Finish, finish, finish. Yep. That's your practical paper. All right, so again, that was 2019. All right, so I'll catch up with other reviews for other paper 5s. So see you soon. Be safe. Good luck. All right.